So I'm just going to let you know what I'm going to talk about in the meeting, and we're really excited about this project that we are developing in the lab. So our research group is very much focused on understanding axon growth during development and axon regeneration in the adult central nervous system. And as most of you might remember from your biology courses, what we are taught is that mammals, adult mammals, are unable to regenerate. And this is really a problem, because this means that whenever a human has a neurodegenerative disease or an injury to the central nervous system, which includes the spinal cord, the axons will not be able to regenerate. And this is a major dogma in the field that has been widely accepted. So something like 6 years ago, a colleague of mine told me that there were these really amazing mice, which are the spiny mice, or acomish, which were capable of regenerating a number of tissues, including the skin and the ear. So the spiny mice are very difficult to be handled and to be kept in a normal facility. And there are only something like 10 colonies in the world of these animals. So completely by chance, we understood that one of the colonies of acomish was held at the University of Algarve, which is in the south of Portugal, and very close to our lab, just like 600 kilometers. So we really started by asking this really crazy idea at the time of whether the spiny mice, beyond the capacity to regenerate the skin and the ear, would have the ability to regenerate the central nervous system and the spinal cord. So I contacted my colleagues at University of Algarve, and we started to try to investigate this question. And really, we were really amazed with the spontaneous capacity that the spiny mouse has to regenerate its spinal cord following a complete transection. So the spiny mice are capable of building a very permissive environment for axons to regenerate in a functional way, meaning that after an injury to the spinal cord, they spontaneously recover a number of functions, including urinary function, sensory and motor functions. So really, we discovered that mammals are in fact still capable of regenerating, or at least one mammal is capable of doing so. So we completely overturned the dogma in the field. And now we're really doing the most exciting part of our work, which is to really try to figure out how this specific mammal can do it. And as it is a mammal, it is very close to other non-regenerative species, such as the regular house mouse that we use in the lab. And what we're trying to do now is trying to figure out which is the best way to make the non-regenerative house mouse like acomish-like, in order to test whether these modified regular mice acquire regenerative capacity following spinal cord injury. Of course, that the final aim of our project is to try to translate these findings to human patients with spinal cord injury. And we're really very excited with this. And there are a number of mechanisms that we already identified in acomish that are really novel, and that's what I'm going to address in my talk in the conference. So it's going to be the first time that I'm attending ISCoS. I had already heard about it through some of my colleagues. I'm very excited to go and to be part of it. And why? The reason for that is that really it's one of the most comprehensive, or the most comprehensive meeting addressing the spinal cord in many different manners. So I'm pretty sure that I'm going to learn a lot, because as a scientist, many times we lose a bit the perspective what it is for a patient to live with a spinal cord injury. So I think that this type of interaction and this type of knowledge will certainly be very, very helpful for me and my team to know in which direction to develop our work. So if you're a first-time attendee or if you're a young researcher, please interact with as many people as they are around, with PIs, with other students, with postdocs, and don't be shy. Let them know of your work and try to get as much feedback as possible.